So as everyone is filtering out and back in, we have back-to-back sessions here. So we're going to go ahead and get right started so we make sure that we maximize our time for each one of the presenters. So I want to welcome everybody to forum number 2, which is titled New Targets and Strategies for Immunotherapy. My name is Justin Balco. I am an associate professor of medicine at Vanderbilt University. I'm going to be moderating this session. And I just want to highlight and thank the speakers here that are in attendance today, and our first speaker will actually be joining us virtually from Cold Spring Harbor. I want to thank the audience for coming and attending, and please, we'll have about 15 minutes for each presentation, each of 3 presentations, with about 3 to 5 minutes for questions. So immediately following the presentation, if you have a question, please step up to the mic and we'll be able to call you. The first presentation today is going to be made by Dr. Mikala Egeblad, who is at Cold Spring Harbor Laboratory. She will be presenting the title, Activating a Collaborative Innate Adaptive Immune Response to Control Metastasis. Dr. Egeblad is a professor at Cold Spring Harbor Laboratory, co-program leader of the Cold Spring Harbor Laboratory Cancer Center program on cellular communication and cancer, and a chairperson-elect of the AACR Tumor Microenvironment Working Group. Her postdoctoral training was performed in the laboratory of Dr. Zena Werb at the University of California, San Francisco. There she developed intravital spinning disk confocal microscopy methodology to understand how the microenvironment influences tumor progression. Her research group currently studies how the tumor microenvironment contributes to therapy resistance and metastasis, focusing on the role of inflammatory cells. She has won the Department of Defense Era of Hope Scholar Award through the Breast Cancer Research Program, the Pershing Square Sohn Prize for Young Investigators in Cancer Research, and the Suffrage Science Award. She's a member of the Scientific Advisory Board for Vividion Incorporated and of INSMED's brensocatib Research Advisory Board. So I want to thank you, Dr. Egeblad, for joining us here virtually. Thank you for being able to be here, and look forward to your presentation. Thank you.
Thank you very much. I hope you can hear me. I want to apologize to everybody for not being able to be there in person. Of course, my family came down with COVID again right before I had to go to San Antonio. So today I'll be talking about how to activate a collaborative innate adaptive immune response to control metastasis. And really the idea behind this is that we have in the tumor microenvironment an immune suppressive microenvironment that helps tumors grow. But if we can change that, we may be able to activate the various immune cells, T cells and macrophages, to work together to combat the tumor at the metastatic site. So we already know, of course, from patients that the tumor microenvironment and immune microenvironment can be both supporting and restraining. There's lots of literature showing, particularly of the myeloid-derived immune cells, that they can promote metastasis and establish pre- and pro-metastatic niches to help the tumors further establish metastasis and grow. And conversely, it's also well known that there are restraining immune forces at the metastatic site. Particularly, it's known from the early organ transplantations from cancer survivors. Then when the host received immune suppressive therapy, sometimes metastasis that had been occult and controlled by immune system started growing out. So what we, of course, is hoping is we can try to move from the supportive to the restraining in a patient. So the way we went about doing this was actually coming from the macrophage point of view. It's well established that there are many different types of macrophages, and simplified, there are inflammatory macrophages activated by bacterial components such as LPS, lipopolysaccharide, or endotoxin, and pro-inflammatory signals such as interferon gamma. And then conversely, there are on the other end of the spectrum, the wound-healing macrophages activated by anti-inflammatory signals, IL-4, IL-13, and TGF-beta. And we know for a long time that these wound-healing macrophages have a lot of similarities with tumor-promoting macrophages seen in the tumor microenvironment. And when I left to start my own lab quite a while ago, after having trained with Zena Werb, I was fully convinced about the role of the tumor-promoting macrophages. But the idea that there were these tumoricidal macrophages that were similar to inflammatory macrophages was very foreign to me. It was mentioned in reviews over and over, but I must admit I didn't truly believe that they existed. So I had a postdoc join my lab, Tim Keyes, And he really wanted to convince me that this was worth investigating. This was what he wanted to do, not the tumor-promoting macrophages. And the way he convinced me was through this particular experiment you'll see here, where he took tumor cells colored in red with fluorescent dyes, mixed them with macrophages colored with fluorescent green protein, and then either left the macrophages unstimulated, where you now can see that they're not controlling the cancer cells at all. The cancer cells over a 2-day period is happily proliferating and growing and filling up the whole tissue area, pushing the macrophages to the side. And then conversely here, he incubated the macrophages with LPS and interferon gamma. And you can see how little by little the macrophages are attacking, killing, eating the cancer cells. And by the end of the 2-day incubation, there's pretty much no surviving cancer cell left. So this assay caught my attention, and I thought, well, we need to do something here. Why is activation of tumoricidal macrophages not a treatment, and what can we do to achieve that? There are many good reasons why it's not a treatment yet. First of all, the macrophages that Tim Keyes had used were from naive mice, so they were not the macrophages we already have in the tumors that we know are polarized to be tumor supportive. We also can't really use LPS in people. It's toxic. Endotoxin is the name for good reason. And then we and others have shown that LPS actually can induce inflammatory responses to promote metastasis in large part by acting on neutrophils. So we could not only be toxic for people, but we may actually do more harm by using LPS. And finally, even if we could get it, it could work, how would we get it to the tumor? So then I had a new postdoctoral fellow, Yuan Sun, that decided to try to see if she could chip away at some of these hurdles and get us further towards targeting macrophages. So the thing we need to try to achieve is to push these tumor macrophages to the tumoricidal macrophages in a safe way. And collaborating with Sylvia Adams at NYU, we started testing different approaches to try to stimulate the macrophages. One approach we used that was successful was monophosphoryl A, MPLA, which is existing as a synthetic version. So it is this little component here of the LPS molecule that is used synthetic form in vaccines, including the shingles vaccine. And so you can get that safely. We then, with Sylvia, obtained pleural effusions from breast cancer patients with metastasis. These pleural effusions, of course, contained cancer cells, which Li Zhuang isolated and labeled with red fluorescent dye here. And then they also contained a lot of macrophages that she separately isolated and activated. And you can see if she just put them back together, the cancer cells are surviving. They're growing a little bit, but not so much here in the patient-derived cancer cells as the mouse cells. If she treat this co-culture with MPLA and interferon gamma, you can now see 2 days later there's not any cancer cells surviving. You have just this little bit of debris. A lot of it is in the macrophages that have been activated. And what is really important here is that you need both MPLA and interferon gamma. Either component alone is not doing anything. The cancer cells survive. The macrophages are not sufficiently activated. And the quantifications over here. So how do these macrophages kill the cancer cells? It's a classical mechanism that had been shown in the literature before. It's through iNOS that is generating NO that then transfers into the cancer cells and kill them. And you can see, again, it's important that you have both LPS and interferon gamma, or in our version, MPLA interferon gamma. Otherwise, you do not get the high induction of the iNOS protein. And you can see also here that this is really the mechanism. If we inhibit iNOS with L-Nil, MPLA and interferon gamma no longer can kill the cancer cells. So this is the great, but it's all in vitro, even if it's human macrophages and cancer cells. How do we get this to work in a real tumor? And to try to achieve that, we started simply with doing an experiment. I must admit, we didn't fully believe what would work as well as it did, but simply doing intratumoral injections repeatedly into mouse tumors. What you can see here is if we do that, we now have expression of iNOS in the macrophages has more than doubled. And again, you need both MPLA and interferon gamma to see this increase in iNOS in the macrophages. More importantly, we can see that this injection actually is controlling the tumor growth to some extent. So the tumors are still growing, but they are growing much slower. Particularly here you see the polyomymality tumor model is very responsive, less so the 41 model. And in both cases, we do also see some response of just interferon gamma alone, probably activation of T cells particularly, but only when we combine the two do we see a strong response. Now, that's great, but as I mentioned, we were worried about whether there would be any detrimental effect on metastasis, whether we would enhance metastasis. Both these models are metastatic models, so we could test this. And to our surprise and delight, what we saw was actually the opposite. When we treated these animals with intratumoral injection in the primary tumor with MPLA and interferon gamma, we saw a very significant reduction in metastatic burden in these animals in both models. And in both cases, there was really the combination treatment that was effective, although interferon gamma by itself also, as you can see, was able to reduce metastatic burden. So that, of course, raises the question, how can a local activation of macrophage reduce metastasis in another organ? That doesn't make fully sense unless, of course, you start wondering whether these macrophages are talking with other cells. To start to get into this, we began by doing RNA-seq of the tumors that were treated either with vehicle injection, with MPLA and interferon gamma mixed together, or with MPLA alone or interferon gamma alone. And you can see really this combination is activating a slew of genes and downregulating also genes that is quite unique to the combination. Each of them alone is not doing anything remotely the same. If we do go-term analysis of the upregulated genes, a very clear pattern comes up. Type 1 interferon signaling pathway, antigen processing and presentation, response to interference, and defense response to virus. And that, of course, really hinted to us that adaptive immunity was important. What we also noticed, but have not yet chased, is that there was a downregulation of genes involved in ECM regulation, collagens and extracellular matrix organization, particularly, were downregulated. So now, with this knowledge, we went back and tried to see what would happen if we depleted either T cells together, both the CD8 and CD4s, or targeted the macrophages with anti-CSF1 receptor antibodies. And you can see in both cases, they had the same effect on the primary tumor. If we deplete either T cells or macrophages, the treatment no longer works. At the same is the case for the metastatic burden. Deplete either T cells or macrophages, there's no longer a reduction in metastatic burden. So we really need both cell types together. How does this work? Are the T cells activated by these injections? So some of it may very well be interferon gamma acting directly on T cells or even MPLA, but some of it we think is mediated from the macrophages, and that again is exemplified by the idea we need both cell types to get the full responses. In vitro, we can see that macrophages are affecting the T cells after these in co-cultures, and uniquely doing so when we incubate them with MPLA and interferon gamma, as shown here by upregulation of CD69 expression. This is mediated by secreted factors. We identified interleukin-12 and TNF-alpha as two critical ones found from RNA-seq analysis. We're also seeing that the macrophages are expressing higher level of a chemokine CXCL9 that we know is good to attract T cells, but of course it's not important for the activation of T cells. So we now have something that seems to work in the mice, but how would that be realistic to translate into patient treatment? And that's something we're still working on, but in the meantime, we were thinking that maybe a way of looking at this was trying to look in another cancer type where we have a good location where the metastasis and the immune cells are all present, namely peritoneal cavity. So we switched to a model of ovarian cancer, metastatic model, using a cell line that is spreading through the peritoneal cavity, and then starting our treatment at day 21 when it's already spread, and continuing until day 48. And you can see that this had really a dramatic effect. This is the bloody ascites for vehicle-treated animals. They were really looking not quite well at this time, whereas the treated animals, we had to flush the cavity with PBS to get anything out and get a little bit of remnant cancer cells. In survival, as you can see, if we do the treatment now starting even later at 42 days after inoculation of these cancer cells, we get a dramatic increase in survival here in the red line, and it's as good as if we start treatment earlier with chemotherapy with cisplatin. But the best survival benefit we are seeing is if we do the cisplatin first in the early time, possibly debulking the tumor, but also likely affecting the immune environment in the peritoneal cavity, and then come in with the implanted interferon gamma treatment. In that case, we actually have mice that survived apparently disease-free for as long as 5 months. So with that, I want to conclude and say that I think what we learned from this study is that the tumor immune microenvironment is plastic enough. We can reprogram it to control both tumor growth, but even more importantly, metastasis. And one approach, of course, is to use implanted interferon gamma. The treatment works because it's engaging both the innate and the adaptive immune system. So the macrophages and the T cells are both required, both for the local effect, but also for the effect on at the metastatic site. How important are these specific factors that we identified in our in vitro cultures? INOS secreted by, activated in macrophages and leading to NO secretion, IL-12 and TNF-alpha in vivo. We don't know that. That's work that's ongoing, but from other literature, we think that particularly IL-12 might be a very important factor to try to get up in the microenvironment. INOS perhaps is more complicated because it can pro and anti-tumor effects. What are the roles of other cells, dendritic cells and fibroblasts in the potential reprogramming? That's also something we are looking at. In particular, as I mentioned, there was a downregulation of ECM genes, which may indicate that there are changes in the fibroblastic environment too. Fibroblasts, as we know now, have a major immune modulatory effect too. And of course, one thing that to me is intriguing, why is it that the chemotherapy followed by the treatment is so effective? Is it simply debulking, or is it that the chemo is changing the microenvironment to make the treatment more effective? And so with that, I'll be happy to take questions. I just want to particularly acknowledge Li Juan's son that was able to really bring this project to the finish line. She's currently looking for jobs in industry, so I grab her. She is extremely careful and diligent, and I highly recommend her. I also want to thank my collaborator, Sylvia Adams, particularly, who has been so helpful in both providing material but also intellectual thinking about how to do these experiments. Ian McNeish for getting us to work on ovarian cancer models, and Phyllis Egimoto working with the biostatistics, and finally at Cold Spring Harbor, David Spector. And so with that, I thank you for your attention. I'll be happy to take any questions. Thank you very much.
Thank you very much for that great presentation. Do we have any questions from the audience for Dr. Egeblad? Over on the right first.
Hello, Matteo Bocci from Lund University. Thank you so much for the fantastic talk. This reprogramming, do you know if there is a preference over resident macrophages versus recruited macrophages? And if so, do you think that the programming could already happen at the monocytic stage, and that would impact also metastatic dissemination?
I think that's a great question, and we don't formally know, so I have a suspicion that it affects both. I think that the reason the chemo is working better is that it is leading to a recruitment of monocytes. We and others have shown that there is a response to the debulking with recruitment of monocytes. And so I think that part of the reason it works better then is that you get more monocytes in that you can affect with the treatment. We do think it also will affect the resident macrophages or the macrophages that are already present, because we've done experiments where we took macrophages from the tumors and treated in vitro to see if we could stimulate them. But formally proving that it's a little hard, we were trying to do it, and then COVID hit us, and we never revisited those experiments.
The middle microphone.
Yeah, my name is Katri Selander from the University of Oulu, Finland. 2 questions. Does the MPLA somehow bind, or is it a ligand for TLR4? And did you look at direct effects of this MPLA on breast cancer cells in the absence of macrophages?
Yeah, so you're right for both points. So MPLA is acting on TLA4. We have that as I think supplemental data, and it does affect cancer cells, but not to any effect on their survival. We did experiments on that in culture, so it doesn't affect the survival or the proliferation of the cancer cells, but of course it does change the gene expression. And I think in the tumors, it's possible that this contributes to it.
Thank you. I think we have time for one more question over here on the right.
Hello, Miguel Quintela from the CNIO at Spain. So my question is about timing. I wonder if you were tracing by any imaging method whether the animals had already established metastasis or not before the intratumoral injection. I ask this because on top of the potential activation of CD8, CD4s, there might be a role of these M1-like macrophages in normalizing the environment, microvasculature, etc. So just to know which of these 2 factors might be more important.
Yeah, no, great question. So we've seen effects both if we do it before we think there's any dissemination, so suggesting that you could be right, that there's effect on dissemination, but we've also done some experiments where we gave the treatment intratumorally and then re-challenged animals with new cancer cells that were labeled with a fluorescent dye, so we could track them and then could see that it actually affected the ability of these new cancer cells to take seat in the lungs. So we think it's definitely an effect on the immune response at the metastatic site, but I will not rule out that there's also an effect on dissemination.
Great, thank you very much. So because we lost a few minutes there in the shuffle, can we give one more round of applause for Dr. Egeblad?